And we have a set of speakers for this session that are going to share with us their knowledge and expertise in predicting in new problems, new data sets, new models, and new methods. And I think we have a great speaker to start this session today, Dr. Charlie Messina from the University of Florida. Charlie?
All right. Thank you very much for the invitation. I'll try to stay on time. If I go over, just 80, please. keep me on time. I also want to thank Tom Tang, who is from Corteva, and he should be somewhere in here, and Lucas and Mark Cooper, who are co-authors in this work. I start with making some assumptions, so I'm not going to discuss that. That's at least my working hypothesis. I truly think that we can reimagine agriculture to make agriculture part of a solution to climate change rather than being the cost of part of the problems, including health and whatnot. Today, they cost about $2.1 trillion, the way we work our fields. And the other one is, I'll make a statement, and hopefully we can have a discussion at some point, but we absolutely have a lack of a framework to think even about climate change and crop improvement. So the questions that I will focus in this talk, one is, do we need to initiate crop improvement programs to adapt to climate change? That if you look at proposals and research programs, that seems to be the case, that is must given. I would like to propose that may not be the case. And if we need, in certain cases, how that framework should look like. So those are the two motivating questions that at least drive part of the talk I'm proposing here. So we are not the first ones to start asking these questions. There were other scientists, recent papers in PNAS, all the papers in Science. They use approaches that I will call it are observational studies, different databases, some of which they have temperature as covariate, some other ones they have a little information about management. Certainly there are many, many assumptions about genetics. The problem with that is that this system is dynamic and is nonlinear and interacting. So as what you see in that time series is the result of breeders working the germplasm for the current management. If farmers figure out the management, how to improve the use of that genetics, the climate may or may not be changing on top of that. So taking a time series and deconvolute without information what are the causes is a kind of a risky proposition and can lead to pretty wrong statements about what was driving what. So other approaches are based on crop models. Christine have an explanation about that. We have a discussion about it. And in this particular case, because we have these delta temperatures predictions, these McKinsey, but there are a million maps that look like that. What we do with these mechanistic models is we can fix genetics and management and ask what are the consequences of a changing climate. And that's what very much did in this study in Nature Climate Change. So they went and they look at how 30 simulation models compare against one experiment, seems to be covering a fairly wide range of temperature. They call it the hot cereal experiment. And the genotype is Jecora rojo. As a breeding community, that you have 30 crop models and one variety. And now we can start thinking about what is the impact of climate change. The conclusion is given, give or take, is about 6% per degree of temperature increase, which the implicit statement here, or logical deduction, is the future is going to be warmer. The models simulate well Jecora rojo. They will have an impact on yield reduction. Therefore, we must breed crops for the future. There's a little bit of an assumption here because the simulations on the impact do not account any strategies of adaptation and breeding. So again, you can see that the approaches we are looking here, they are quite incomplete at best. What we really want to have is a data set that is run over time, whereby you can have the same genetics with a changing environment and the main drivers of management fixed. So what we can do is we can start taking derivatives of yield with respect to genetics, to environment, to management, and then we can start drawing some conclusions about what is happening there. I don't know if you know how many experiments are like that have been run for 30 years or so, but there's one run by Don Dubix. The first experiment was run in 1972, but there's a fairly long series of experiments run from the 1990s. So that will give us almost about 30 years worth of data that we can use to start deconvoluting what happened over the time. So one thing that also needs to happen for that study to provide any insight about the opportunities or need to do breeding for climate change is that the climate has to change at the same time as breeders were working their germplasm. And this is what happened in the Central Corn Belt. This is data from NOAA. It's a regional, so it's not point or Ames. So it's a pretty robust picture of the changes in temperature, precipitation, and amplitude temperature from the 1900s, and should be pretty obvious that the precipitation increased, minimum temperatures increased, and the amplitude temperature decreased about a degree or so. So it's pretty significant. If you look at the shaded area, that marks the beginning of the pioneer breeding program. So essentially, we have a running experiment here where breeding and germplasm has been evolving during changes in climate, and of course management as they optimize. So now you have an experiment we can interrogate and partition genetics, environment, and management components, and we have an experiment, at least the longest and continuous breeding experiment with well-defined objectives while the climate was changing on us, and it was significant. Significant, it seems important. 1 degree temperature is quite massive. So with that, then, what we would like to do is select from that data set a set of hybrids that were maintained constant for the longest period of time. And in this figure, the one below, the one is the top is just a cartoon to show you and remind you of the changes in the climate. Each black dot represents one successful hybrid as was defined by Don Dubik and Cooper and Delos, and I can remember who else was part of that discussion. Maybe the gentleman on that corner has something to do with it. But nonetheless, each dot represents a hybrid that farmers define as a successful hybrid. So you can see that all these hybrids were commercialized, and just as a background, they were commercialized and produced at different times, and you can see the reduction in the environment. So with that, we can select hybrids that were commercialized between 1930 and 1990, so we can calculate some genetic gain over 60 years at least. And then what we can do is we can make predictions of what could be or what can happen between 1990 and 2020, because these hybrids were not produced during this era of the remaining time in that time series for the effect on genetic gain. So to recap, the ERA study has about 50 genotypes be commercialized between the 30s and the 90s. This is a complete set that we maintain constant over time, and we ran this experiment over 30 years in Ames in 3 locations. And on top of that, because part of the Don Dubik's hypothesis is that selection imposed by breeders were imposing or leading to improved stress tolerance, they have 3 treatments of management which underpin major changes in productivity in corn, which is plant density. So now you have the environment is changing, we know the genetics, we know the environment, so we can partition, and then what we can do is calculate for each year the rate of genetic gain. And with those genetic gains, now we can condition upon low density, normal density, and high density. So each of these points, the year on the x-axis now is not year of commercialization, is calendar year. So what is plotted is the rate of genetic gain, meaning if you see there something about 2.5 grams per square meter per year, what that means is every year, even if that one is greater than 0, there was genetic gain, regardless of the change in the environment. When you add the increase in density, so now you have the management optimized to maximize the growth or the genetic potential of that material, you almost double the rate of genetic gain. And you can see there's a variation, yet some years they do better, some worse. We are not sure what is that, but definitely it's about 7 grams per square meter per year. And if you look at the high density, now I think you can start seeing even the genetic gain is accelerating. So climate is changing and the genetic gain is accelerating. So do we need to breed for climate change? So I will leave that picture and move to a little discussion of recap. Genetic gain, let's assume, is these almost isolated plants. We have a G by M as we optimize the management, and with the changes in the environment, you can even see almost an acceleration in genetic gain in maize, in the Corn Belt. So again, I will emphasize this so you remember that the rates are greater than 0. Even at the worst case scenario, they're greater than 0. So what that leads us is going back to these approaches that they're advocating that genetic has nothing to do with genetic gain based on trying to deconvolute this time series based on a little bit of management and making assumptions on the hybrids that were commercialized over time is quite incorrect. We have like 17,000 records over 30 years of data that suggests that Dr. Grassini is not right and this thing is probably should be retracted. There's another one that was published in Science, and they said that sensitivity to drought has been increasing as well. We have some discussions about that. And as you can see, genetic gain was increasing over 30 years, including the drought of 2012 and of course was the 2013. So when you try to deconvolute these things on management, environment, and genetics, you can start driving the wrong conclusions because you don't have the data and the right experimental approaches to justify the hypothesis that you put forward. So again, another evidence that how risky is trying to deconvolute this series without information and how much you can misinform policy as you move into the future. So going back to my first question, do we need to breed for climate change? And the answer is, if we keep the breeding objectives the same for the Midwest, the answer is absolutely no. What happened is the rate of change in genetic gain in climate was slow enough for the breeding programs to catch up with it. And what happens, and I will try to elaborate on that, is the selection environment were predictive of the performance environment, so the testing environment. Therefore, it was slow enough, and as long as the breeding objectives remain unchanged, what we need to do is keep doing business as usual, because that is what is working. Actually, it's accelerating for whatever we are doing, probably technology and some agronomic management. But now what I will suggest, if we want to now take agriculture and use it as, or consider it as part of the solution to climate change, we need to reconsider now the breeding objectives. And part of the story of breeding for climate change is not about increasing yields, that's a given, but increasing yields while reducing nitrogen emissions, regeneration of water resources, increasing carbon, delivering ecosystem services. And you can add some things to the list as Jose, Christine alluded before. So in this context, I think the breathing for climate change is not because the environments are going to be warmer, but because we want to change the breathing objectives so we can sequester carbon and whatnot. Different story. So let's do a couple of hypotheses why we see what we see in this time series. And there are two hypotheses. One, you saw the precipitation was increasing, and the other one, I show you amplitude temperature was decreasing. And if you talk to maize breeders, they will tell you increasing minimum temperature is the worst thing that can happen to maize, and we'll get there. So if you go to this, first looking at the rainfall pattern, what we can do to test that, and we'll see here is about 50 millimeters of water. So if you think about 500 is about the mean or the centroid of that heat map. That heat map is the US corn TPE represented in a gradient of evapotranspiration and grain yield, so that's including frequencies. So if you take that centroid, about 500 millimeters of water is what rains typically in the central corn belt. 50 millimeters, 10%, that's huge, particularly if you go to Western environments. So now we have this simulation, if you will, TPE. We have billions of simulations here for different genotype, management, and locations all throughout the US. And you can see if we assume that this center is about 500 millimeters, you add another 50 or so, you can explain the change in genetic gain by looking at the impact of water in the productivity of maize, at least over a couple of billions of simulations. So what else may be happening here? And I would like to bring to your attention the light use efficiency, which is a balance between respiration and photosynthesis. And this is a paper we just published in Journal of Experimental Botany after like 10 years of experimentation in Corteva. That's a different story. And what you can see is that the light use efficiency, whether it is because reduced transpiration or improved photosynthesis or improved nitrogen dynamics, we don't know, is been increasing since the 1930s. And on average, this is about 0.3% per year, which doesn't look like a whole lot, but in 100 years of reading, that's 30%. It's 1/3 of the RUE over time. If you think light interception is constant or maximized by optimizing plant population, 30% of the yield is driven by this, which is a single most important trait I bump into. So now we can start asking the question, what are the environmental factors that affect the radiation use efficiency? And of course, temperature is part of the transpiration. Minimum temperature is part of that. You can see increase the temperature, light use efficiency goes down, probably because of respiration effect. But the amplitude temperature, look at that fit. There are not a lot of points in there, but underneath each point, these are 5 different years and locations between Woodland and Viluco, Chile, and there are about, 150 plots in there. So there are 50 hybrids, like 20 hybrids by the replication. So there's a fair amount of data behind of each of these points. We have quite a bit of confidence that they land where they are, so the regression, I think, has a merit to consider this. So given there was a decrease in amplitude temperature about almost 1+ degree, you can see the effect on RUE that change in amplitude temperature can have. So what I think it had been happening because of this smooth reduction in amplitude temperature, the environment was pushing and or creating a more challenging, increasingly challenging environment for selection, but because there's a smooth negative trend, they was pretty predictive of the testing environment. So genetics, sort of selection, push the thing up, and the environment keep pushing it down, and genetics keep pushing it up, and so it was climbing slowly the ladder. And over time, it's explained that 30% yield improvement just in a single trait. There may be some other things that may lead through radiation use efficiency, plant growth, kernel numbers, and yield. So there are more to it, to the story, than just the RUE, but I just want to give you an example of one trait, why we see yield or genetic gains which even are increasing despite of climate change, because the predictive environment or selection environments are predictive of testing environments. And that sort of takes us almost to a pivot point of what sort of framework do we need to consider before we start funding projects for adaptation to climate change in the absence of any data. So we have the match between the TPE and the multi-environment trial. So this 0 match to 100% match, and the same thing over there. Within current and future, sort of multi-environment trials and future TPs, and the Y is the, if you will, genetic variance. Assume is 1 over there, and 0 over there. So there's a mismatch between selection and testing environment. Your covariance is 0, pretty straightforward. You get the wrong frequency of boron iso selections, your covariance is decreased. However, when there's crossover interactions, like they may occur, you may end up with negative impacts on selection. So the US Corn Belt is right there because that's what I show you. The changes in environments are predictive. So that's why you have a pretty high covariance and drive genetic gain. The big question, or the elephant in the room here, is what are the other experiments and experimental data for the other crops and the other geographies? How many other era experiments we have that have been run for 30 years that will enable us to write conclusions about, do we need to breed for climate change for soybeans or wheat or whatever that we choose to do so. So the question is, we need a framework, we need methodologies to answer that question. And the problem is, we can't get too fast in the breeding process for future environment, because we may run the risk of starving the current population if there's a mismatch between the two. If we start too late, we'll starve the future population. So that's why I think we need a new framework or rethink, bringing the breeding perspective to the discussion of how we are going to adapt to climate change. So part of the story, I think, and this is my views and part of my speculation, what the elements we might want to bring together is bringing scientific understanding and statistical learning. Christine called it crop model whole genome prediction. So essentially, the framework she put forward is something that can help us deal with that. Part of the story is we need to have an understanding of the percent of genetic variance in this gradient of evapotranspiration, and to understand the complexity of the traits, to be able to predict that complexity. All this area you can see is trait by trait, so almost in the Midwest for maize, 25% is explained by a few traits and the drought, the 500, 50%, at least in this simulation. Most of the yield is determined by interaction between different traits. That's a difficult problem to predict. And it's even worse to make things even more complicated, as we think if we're in the West, Colorado, 200 millimeters, and the environment changes to E2, there's not a whole lot of difference. So the predicting and testing environments are going to be about the same. If you change the E1 to E5, range quite a lot, the combination of traits is quite different, and you have to cross all throughout this complexity of different traits as you start moving that germplasm across the different environment types. So depending on current climate and future climates, we can have all sorts of different interactions that we may have to deal with. And we need a framework to be able to predict where we are, in which circumstances, and what breeding solution is needed. At the end, what we would like to do is, using the approach that Christine explained, generate yield, evapotranspiration, and start mapping the hybrid. So if this is your current climate and your future climate, we can make informed selections on how we are going to use the water, the food, and use make decisions for carbon. Again, this is going to a little bit of that decision support, but hopefully we'll start getting into different ways of making decisions, not just on yield, but some of the other ecosystem services. And if we trade the models the way Christine described it, we can get there. And finally, I think this framework is going to be necessary to apply, because today we are developing these genotype biomanagement technologies, essentially genotypes by plant population, considering yield, maybe morphology and some physiological traits. But as we think about nitrogen, maybe we can measure things in the field, water, maybe carbon, we can't. So essentially what we need to do is generate these models that can give us an idea of what are the consequences of our action in the future environment, and enable us to make decisions not just as a multiple traits, but in these multiple dimensions. So to conclude, hopefully I convince you that we need a framework for climate change. I hope I convince you breeders need to be part of the discussion, otherwise we are going to make really bad recommendations for policy. We need to consider all the levers, genetic and agronomic, that will deliver that value for society. Prediction frameworks need to help us predict these emergent phenotypes to navigate these landscapes. And finally, I will advocate for this integration of statistics and dynamical models, so we can borrow information through genetics and physiology and biology in our data sets. So with that, thank you very much.
Thank you. We have time for a couple questions.
Thank you. It's good presentation. My first son name is Charlie, and I tried to persuade him to be the breeder, but he never. He's the IT man in New York, CEO of the NextGen. But my 50 years of experience with corn breeding in Hawaii, Illinois, and Pioneer, all this, I think under climate change, breeding target must be by using QTL, not single gene. That's my comment. So anybody, if you want to nearly spend too much time for single gene, the mutation will come and you will collapse. So that's my comment.
I think it fits within this framework Christine shows. So I think your comment is highly appropriate. Not sure if QTL, but certainly genomic prediction or a combination of genes will underpin performance. Certainly that was happening with Aquamax. And there's another paper that shows that after 20 years of breeding for drought tolerance, you can make that improvement without having to get to single gene solution. So no disagreement there.
One more question?
Very nice talk. Your analysis suggests that the way breeding's been done so far, it's the selection environments are predictive of the target environments, so we're keeping up with climate change. I'm not sure how much genomic selection was used during the time period that you analyzed to know, but can you speculate? I mean, genomic selection, the goal is to accelerate that rate of change based on past performance. Are you worried that there could end up being a disconnect between the timescales involved there?
I can speculate all day, but I think the last years on that high density, we're essentially showing that the modern hybrids take more advantage of better environments without having the penalty on the bad environment, is the result of the use of technology. And I didn't show that, maybe Tom can comment. If you look at the the post-1990 hybrids, which is the smallest set, it's the same. So I think genomic selection, what is enabling you is just increase the size of your breeding program. You need to be mindful of how quickly you're going to run out of genetic variation. I'm not sure, but certainly will help you catch up faster as the changing is changing on you. So as long as you have a fairly large and diverse germplasm, it shouldn't be a problem to navigate the landscape. If your genetic pool is very small, you probably are hinting at the right thing. It's just going to get stuck in a local maximum and you will never be able to get out of it, right?
Okay, let's thank
I-
Dr. Messina once more.